Yes, we are live.
Hello everyone, good evening. I am Sandeep Singh. On behalf of NIBMG family, I welcome you all uh, to this webinar series. And this series is going to be live on YouTube, and there's all the participants are allowed to write their questions in the chat box so that we can take up the questions and put in front of the speakers. So just to introduce about this webinar series, we have initiated this series to celebrate India's festival of being at 75 years of independence, which is Azadi Ka Amrit Mahotsav. And this series is going to celebrate India's pride and self-reliance in the field of genomics, health, and well-being. So this is to showcase our strength in the field of biomedical genomics, the research and the strength which we have gained. And because of this technology which we have developed, and we would like to showcase India as well as the world that we are among the leaders, and we are going to lead lead world through India with the help of this research. So in this webinar series, we have our distinguished speaker today, who is Professor Shantanu Chaudhary. Professor Shantanu Chaudhary is from CSIR Institute of Genomics and Integrative Biology, New Delhi. After his PhD in the chemistry from IISc Hyderabad, he is trained in molecular biology at the University of Nebraska, USA, and the Max Planck Institute, Marburg, Germany. In 2002, he moved to his current position at ISCB, New Delhi, where he established his own laboratory. He has received various prestigious awards, including Shanti Swarup Bhatnagar Award, and in 2012 in the biological science. And he is a senior research fellow from Wellcome Trust DBT India Alliance and serving as editorial board of Journal of Biological Chemistry and Human Genomics. He is also elected as fellow of Indian National Academy of Science, India. And his research interest is understanding the effect of DNA secondary structure on gene regulation, cancer metastasis, and develop intervention and strategies to restrict the spread of cancer. Today, he is going to talk about glioblastoma, which is, as you all know, is a devastating disease. And Professor Chaudhary is going to highlight his contribution to this field, and he is going to share his story about his contribution to making India self-reliant in the field of genomics and glioblastoma and disease biology. So without any further delay in this webinar, I request Professor Shantanu Chaudhary to share his slide. And as I have already mentioned, Professor Chaudhary, that this is a online seminar, so a lot of audience have joined other than from NIBMG. We have advertised this about your talk on the online platform. So many speakers from many audience from outside has already joined on YouTube, and they are listening live to you. And all the audience on the live who have joined us, please write your questions and comments on the comment box so that I can put it across to Professor Chaudhary. So Professor Chaudhary, please join the webinar.
Thank you so much. Thank you so much, Sandeep, and thank you, PMG, for this opportunity. And indeed, it is a great honor to be part of this celebration. And I'm going to start sharing my screen, and hopefully
Yes, I can see your slide. Yes, now it is full screen.
You're muted.
Your mic is muted.
sorry. Okay. Yeah. Once again, thank you very much for this opportunity. And like to sort of begin with showing a beautiful painting by Peter Paul Rubens in a famous Baroque style, as it is called, in the 17th century painter, who is talking about a mythological character, uh, Achilles. And Achilles in Greek mythology and later on also has become a, um, somewhat like a rock star in the field of wars. So he was a brilliant warrior apparently, both in terms of strategy and skill, and was all quite invincible through the several years of his that he lived and fought. So the story goes like, he was prophesied to be to die young. So his mother was very concerned, and she took him for a dip in the holy river, which was supposed to give immortality and thereby save him from the prophecy that was made against him. While doing that in the river Styx, she held him by his heel, and therefore the waters did not touch the heel. So goes the poem, or the famous poem by Homer in Iliad, Homer's Iliad, where he talks about this. And therefore, this water, as since it didn't touch the heel, later on in the famous Battle of Troy, the Trojan War, I think he was actually an arrow was somebody knew about this apparently, and arrow was targeted towards his heel. That was the end of a really invincible warrior. In the context of today's story, there are two important aspects that I would like to take from this very interesting story, which has become a metaphor of invincibility. And if you think about cancer as this robust and strong system of an almost invincible disease, then one can understand why how Achilles heel, or a weak point in this otherwise robust system, makes sense. And figuring out such a weakness is probably a very sought after interest, or really massively followed angle of research, that is about how to find out this weakness in a very layered and almost invincible system, which we term as cancer. So once you look into the molecular processes, you are continuously looking for the Achilles heel, or the weakness in this molecular strategies or the molecular processes which contribute to cancer. So I would be talking about one of those that we sought to find out and we worked on. And the second aspect that this metaphor brings to the forefront is that once you understand, then you can also design strategies much like that arrow which was targeted towards Achilles' heel. So much like that arrow, once you understand a molecular deficiency or a molecular process that has gone wrong, how does one target the heel of the process, or the Achilles heel of the process, so to say? This would be a two intertwined themes that I'm going to sort of talk about uh, in the next half an hour or so, and hopefully bring to the fore by the end of this talk how we strategically focused on this, and how we could sort of understand this in the context of glioblastoma, which is probably one of the most aggressive cancers that crowd there, where which is very difficult to treat, and also the survival percentage, or the disease-free survival, is really very pathetic. So one it, it is an invincible disease in some ways. So before I get into the thick of the story, let me introduce a few characters which would run through the story. So one of these is telomerase. Interestingly, it is the only protein that makes telomeres or synthesizes telomeres. And we know how important telomeres are. It is spoken of very commonly as a barometer of your health. Longer the telomeres, better, more healthy you are. Shorter the telomeres, less healthy you are. In a very common way, it is spoken of like that. These are the end of chromosomes, linear chromosomes, which are protected by a complex of DNA and protein. This complex of DNA and protein is called the sheltering complex. A major factor in this sheltering complex is telomerase, and this is the protein which synthesizes telomeres. As and when these telomeres get shortened, they are made back to a certain length, and it is very important, therefore, that is, this telomerase is controlled. Why? The reason being that with every replication, every cycle of that the cell divides, the telomere shortened by about 400 to 450 bases, and this is very critical for life. Reason being, as in normal cells, as telomeres, as the cell grows and divides, telomeres are supposed to shorten. This is the paradigm. This is the rule. However, in certain scenarios, and the reason for that is, they reach a critical length, and therefore beyond that length the cells are programmed to die, which means that if telomeres are below a certain critical length, cells are supposed to perish and new cells are supposed to come take their place, and life moves on. However, there are scenarios. In certain scenarios, what happens is, instead of shortening, after a certain period the telomeres do not shorten anymore. And the reason being, this protein that can make, that we just talked about in the last slide, the protein called telomerase, which is the only protein that can make telomeres, it is supposed to be shut down right after 6 to 8 weeks of a gestation period, because it is important that normal cells have very limited amount of telomerase, therefore telomeres are not synthesized, and the natural law prevails. Except in scenarios where by some mechanism telomerase is reactivated, and once telomerase is reactivated, cells forget how to show, to keep their telomeres shortened, and the telomeres get maintained. Maintained means the cell doesn't know how to, it doesn't move to, the triggered cell death doesn't happen, and therefore what happens is cells become immortal. Immortal cells, in another sense, is what we call as cancer. And therefore, why telomerase got reactivated is a very important aspect of studying cancer, because it is one of the hallmarks which leads to the initiation of, or the malignant cases, when cells become become immortal in some ways. So in the world of telomerase, there was a big explosion in the year 2013, not too many years back. While the whole world was looking at exons for mutations, this back-to-back papers showed that there were a few mutations which were non-exonic, not in exons, but in the promoter of telomerase. And these mutations, all of these mutations were G2A. I'm highlighting 2 out of 5 because these 2 contribute to almost 70% to 80% of issues with telomerase mutations. And these mutations are about 124 bases or minus 146 bases away from the transcription start site. And what these papers suddenly showed, that these 2 mutations are found in about 71% of melanomas, brain cancers, skin cancers, metastatic melanomas, primary melanomas, urothelial melanomas. So suddenly there were some promoter mutations which were being associated with the clinical manifestation of this disease. A few years later, in 2016-17, there was almost every cancer that people had looked at and found that these mutations were missed earlier, ranging from anywhere between 20 to 25% to almost 70-75% in glioblastoma and melanoma. People were seeing that the prevalence of these two mutations, point mutations, both guanine 2, always guanine 2, guanine changes, G2A changes. I'll come to in a minute why we were attracted, almost perplexed when we saw this studies coming out. Uh, but the fact remains that these two are now very sought after mutations. If you have these mutations and telomerase overexpression, there are ways people are trying to figure out, how to look at what to do about it. And the rest of this uh, talk will bring a lot of this into the forefront of what we thought about this and what we did about these two mutations. And that's why I termed this as precise intervention, because these are precise issues and need tailor-made interventions so that only that issue is taken care of. And hopefully I'll be able to convince you that we did something in that region, in that area. A little closer look at the telomerase control. I spoke quite a bit about why telomerase control is so important. Now, control, as you know, of a gene is mostly in the promoter. So let's focus on the promoter. And what I want to highlight in the telomerase promoter is this letters in red and black in the lower half of the slide, which is showing that in the promoter somewhere between minus 100 to minus 170 or minus 180, the sequence you see is full of guanines, GGG, arrays of guanines, interspersed with sometimes C, sometimes A, sometimes T. But the remarkable feature is this GGG repeats. And these mutations that we talked about, the substitutions that we talked about, were actually G mutations which were within these arrays. So therefore, let me talk for a few minutes about what is GGG clusters, what are GGG repeats, why are GGG repeats so interesting to look at from a very different angle. I'll have to sort of throw back almost 15 years and go and talk about some of the stuff that we started doing long back, which was studying this GGG repeats and what the GGG repeats do. And they form a very beautiful bookshelf-like structure. If you throw GGG repeats in a test tube, you will get that. But more interestingly, telomeres, the end of our chromosomes, it's about generally 4.5 to 5 kb length before it starts decreasing with every cycle. And the only sequence, if you have cared to look at that, is G3-TTA, GGG-TTA, for 4.5 to 5 kb, 4,500 to 5,000 bases of GGG-TTA at the end of each chromosome, which means that there are both ends, end of P arm and the end of Q arm, both have only G3-TTAs. And when people started wondering about these G3-TTAs, the first thing people wondered were actually chemists. And this is 1980s, 1960s to 1980s, when they started looking at guanine self-assembly. And what was noted and very well studied now is that if you take a millimolar solution of guanine and keep it in a test tube, after a few hours you will not be able to shake it out. It has formed a gel. This happens because guanines self-assemble and form this beautiful tetrad structure. So 4 guanines at 4 corners, and sometimes potassium or sodium helps by making a perfect little hole where this ion can go in. And this GGG self-assembly also drove the factor that if you have a sequence of GGGs now, and you consider out of this sequence that I have written in the middle of the slide, 3 Gs NNN, 3 Gs NNN, if you take 1 G from each of these triplets, quartets of GGGs, and you will form one tetrad. Take the second Gs and you will form the second tetrad. Take the third G and form the third tetrad. In the process, what you have done is you have intertwined a sequence unto itself into this bookshelf structure, which has been studied very well now and is known as the guanine quadruplex or G4 motif in short. In other ways, it is a sequence which can fold upon itself to form a structure, which is known as the quadruplex structure. We become very a little clear a little later why I am going so much into this structure. And what we wanted to do several years back was try to ask, are these GGG, these patterns of GGGs, and it was a fairly innocuous question at that time, are these patterns of GGGs only limited to telomere ends or do you find it across the genome? Cutting a long story short, what we have done here is, this was about 15, 17 years back now. Uh, what we did was we asked, and then once we found these GGG arrays, we mapped them. Here, every promoter in every row here is a chromosome, and every promoter in that chromosome has been, every transcription start site has been bioinformatically tied at the center. So all the transcription start site in a single chromosome is right there on row 1, then the next chromosome 1, 2, 3, 4, and so on. Just to give a sense, what it tells us is these GGG repeats, which were supposed to be only in the telomeres, we suddenly found that these were not only, they were present very much across the genome, and they were not randomly distributed. There was non-random distribution. That is, they were present mostly in the near promoters. So as if when you are upstream, you are going up, and when you are downstream of the promoter, you are slowly going downhill if you are counting these GGG structures. So we proposed that these GGG structures or these G quadruplex structures might have something to do with uh, gene regulation. And then we, of course, went ahead and sort of proved that these structures are important for gene expression because of this non-random distribution that we see. And it was true for not only human, mouse, chimpanzee, rat, and more than thousand, more than we analyzed almost every bacteria out there, every sequence, every genome sequence that we came out, it became a hot area in the lab to sort of go and figure it out because we had this algorithm going. So we found that this is true across the universe. In any organism, plants, archaea, you have this structures or the sequence which can make such beautiful structures very prevalent in promoters. And we went on to show actually that this was not only a decorative piece in the promoter, but had a lot of functional value because we discovered the first transcription factor which could bind to a non-duplex DNA. We call it non-duplex, obviously, because where the structure forms, the duplex form doesn't exist anymore. The other strand hangs below and stabilized by single-stranded proteins, as is very well known now. So in the quadruplex part of it is bound by transcription factors. The first one we showed in 2009 as a factor, and after that, I think about 12 transcription factors are now known specifically to bind to quadruplexes and have quadruplex dependent functions. And we use different molecular methods to show that this is a quadruplex dependent transcription factor. Without the presence of the quadruplex, the transcription factor is not as functional. So in a very, briefly over time, so we went ahead. First was the bioinformatics part of it. Then we used, once we knew that these were sequences, DNA sequences, which formed specific structures, one could, use small molecules which are targeted not towards the duplex, but towards the folded structure, and show that, this folded, this small molecules or G4 binding small molecules, small ligands, could could change, alter transcriptomes. Then obviously a lot of work in terms of, in gene regulation, different genes we picked up, and we showed that these transcription factors, at a molecular level, these transcription factors are important for quadruplex binding. And before we come to today's story, so this is the background from where we were coming. And in 2013, we noticed this telomerase promoter with GGG repeats, which can very well form quadruplexes, and mutations within the GGG repeats. And obviously, with the background that we had, the question that became a hot topic in the lab is, what if these G mutations are such that they are disrupting formation of quadruplexes in the promoter of telomerase. And with all the armor that we had till then about studying such things, studying this aspects, we went deep into this problem. So here again is the GGG repeats on the telomerase promoter. This is exactly how the telomerase promoter looks. And you can see that this GGG repeats are very well set to make those structures. And obviously when you take it out and put them in a sequence, take this sequence out and the fragment of the sequence in a test tube, you definitely find that it forms this structure. So we said that yes, this could happen, and therefore when in the G2A repeats are the repeats, if the guanines are taken away from that stabilized bookshelf-like tetrad structures, if one corner of a bookshelf somehow falls, the bookshelf falls, right? So that is exactly what we thought. And we also thought that if this is the case, can we intervene? Can we do something about the destabilized quadruplexes in the G2A mutations, which are so frequently found and so devastating in certain cases? So this is what we call as the Achilles heel, and our attempt to how to point an arrow or how to target an Achilles heel becomes the rest of the story today. So telomerase reactivation, and as I said, the sheltering complex, and in the sheltering complex there are many factors which protect the telomeres, and we were interested in one of the sheltering complex proteins called telomere repeat factor 2. So why telomere repeat factor 2? Well, again, a little bit of a backstory. This is much more recent than the G quadruplex story. So TRF2, we noted that TRF2 for 40 years has been was studied as a factor which can only bind to telomere ends. In 2011, a paper showed that there were about 120 sites outside the genome. By that time, we had our own data, unpublished data, which we were trying to validate more before publishing, and we knew that the sites were not 120 or so, but much more than this. Later we published this in 2019, where we showed that not only 120 was probably an artifact of the data, more than 20,000 sites of TRF2 were found across the genome. And we also showed that this is not only the story. This 20,000 TRF2 binding sites across the genome, also the amount of TRF2 that was bound across the genome is a function of how long or short your telomeres are. Because TRF2, if you have long telomeres, there's a lot of TRF2 sitting on the telomeres. As the telomeres shorten, there is more TRF2 available and it binds more across the genome. And we also showed that TRF2 was a transcription factor. And this transcription factor, because the binding of the factor across the genome was telomere dependent, resulted in transcription across the genome, which was telomere dependent. So we started showing experiments and publishing work showing that TRF2 is a messenger of how short or long telomeres are, because by sitting across the genome, which can transactivate genomes or repress genomes, depending on genes, depending on how long or short the telomeres are. So we call that telomere length dependent transcription and epigenetic modifications in promoters remote from telomere ends. And this actually is a very interesting story, because so far people had noted that the impact of telomeres is only felt up to 10 MB subtelomeric regions. This work showed that that is only part of the story. The impact of shortening of telomeres is felt much beyond 10 MB, almost all across the genome, because of this simple partitioning issue of TRF2 binding to telomeres and across the genome in the copious amounts. But again, so these two things were then covered by Professor Semia from Japan, who talked about these two papers and said that these studies bring TRF2 from the wings to the center of the chromosome. So it's a very interesting, very short 1.5 page, very interesting read about how TRF2 finds a second home after so many years. So this was also the time when, very active time when two very enthusiastic people joined the lab from two different parts of India and took it upon themselves, Shalu and Anand, to study all these questions that we were just talking about. So if there are quadruplexes, we have already seen that quadruplexes bind to TRF2 across the genome, and then there are quadruplexes present in the telomerase promoter. So the study sort of started they sort of started asking questions about what happens to TRF2 if it binds to the telomerase promoter and to quadruplexes. Then what happens? If that is the mechanism, in that context, what would be the role of the G2A mutations, which are clinically seen so much? So we had this chipstick sitting in the lab. They focused on the telomerase promoter, and they did find that there was the TRF2 binding site, binding motif, which is basically a G-rich motif. They found that, and then they started asking very routine molecular biology questions. Where is TRF2 binding maximally enriched in the telomerase promoter? Telomerase for short is also known as H-TERT. So sometimes in the slides you will see human TERT. So human telomerase reverse transcriptase, that is what TERT comes from. So where is TRF2 binding maximally enriched? Is it true for both cancer or normal cells? So this is the telomerase promoter. And what we noted was we made primers which spanned to almost 750 bases upstream of the transcription start site. And we studied cancer cells and normal cells and looked at TRF2 occupancy. As you can see that in fibrosarcoma HT1080, substantial amount of TRF2 occupancy is there in the promoter, almost going up to 750 bases upstream. This is true for colon carcinoma HCT1166 also. And they also found, Shalu and Anand, they also found that this is true for normal cells, MRC5, HEK293, and we studied a couple of other cell lines also, that there is TRF2 occupancy in the telomerase promoter. So this is what I was talking about. TRF2 binding is there all over the telomerase promoter, almost to 600 to 700 bases, and it is there in normal and cancer cells. So binding is fine, but does it affect telomerase regulation? Promoter activity, telomerase activity. So these were also questions, and these are different cell lines where we are looking at the H-TERT gene expression. And what we see is that when we silence TRF2, these are after silencing TRF2, we see that H-TERT expression goes up in different cell types, which means that TRF2 is a repressor of telomerase expression, which is very important in the context of what we are going to talk about today. But this experiment showed that TRF2 very consistently repressed across cell lines the telomerase promoter. So TRF2 silencing de-represses H-TERT gene expression. This is basically the same thing that we saw in the last slide in a different kind of experiment, immunofluorescence, which basically gives you single cells and tells you what is happening within single cells. Do you see upregulation? We did see that on silencing TRF2, telomerase expression did go up. On a molecular biology, as a molecular biology student, I think it was also important to understand, yes, TRF2 binds to the telomerase promoter. Yes, it is a repressor of telomerase promoter, but how? What is the effect on chromatin conformation? Because that is something that you would expect from a transcription factor, right? So here these are some of the experiments which we focused on 27Me3, which is a very well-known silencer, a canonical silencer, the first one actually that you would look into in most cases. And we did see that on silencing, the 27Me3 occupancy across the promoter falls. And that is probably why you see activation on silencing of TRF2. So TRF2 therefore turns out to be a repressor by making sure that the 27Me3 remains methylated, because on silencing you lose 27Me3 from the telomerase promoter. And so 27Me3 is known to make closed chromatin. So closed chromatin, because of TRF2 is one of the ways by which TRF2 keeps it closed. So when you silence, you see active chromatin, but when TRF2 is there, you have repressed chromatin. That is the basic message from this experiment. More importantly, between TRF2 and 27Me3 methylation, there has to be some methylation. Something has to be there which methylates H27Me3, because TRF2 is not known to be a methylating agent, right? So the canonical repressor complex PRC2, which is very well known as a 27Me3 methylation, happens many times through this complex of proteins called the PRC2 complex. EZH2 is the methylating agent. So there was a gap. We knew TRF2 is there. We knew that TRF2 is responsible for 27Me3 methylation, but we did not know what lied in between. So we suspected whether this complex, this canonical complex, is there. And we started doing experiments. Shalu and Anand started doing experiments. And one of these is the EZH2, which is one of the prime components of this complex. So we focused on EZH2, and they did see that when TRF2 is silenced, the EZH2 occupancy goes down on the telomerase promoter, and that is true for both HTA108 cancer and normal cells. I'm just keeping this as representative. We did this in 4 or 5 different cell lines. So TRF2 recruits the PRC2 complex on third promoter, and that leads to 27 trimethylation, thereby repressing TERT. So we had at least 3 components of this system in place, and that is TRF2 sitting on the promoter, TRF2 recruiting the PRC2 canonical repressor complex, and this methylating 27Me3 to keep the telomerase promoter in a closed conformation, such that telomerase, which is important that it is kept closed throughout the duration of the lifetime of a normal cell. And this is probably one of the primary mechanisms that holds the close conformation on telomerase, because overactivation or activation of telomerase in the span of a normal cell, as we talked right in the beginning, can be so detrimental and so devastating. So now, once we had understood this, and this is again summarizing just what I just said, that it comes in REST and TRF2. We showed that TRF2 REST. I did not go into the REST story. REST is an RE1 silencing. So we saw that it requires RE1. TRF2 recruits RE1 silencing factor REST. REST recruits the PRC2 complex, and that PRC2 complex brings in the 27Me3, and this is what keeps the promoter in a closed conformation. Now, what is the role of GGG repeats in this? So this is what I said, 2 non-coding point mutations which were found in 80% of glioblastoma, 70% melanoma, 68% bladder cancers, and in total about 40% of all cancers from many studies. So one study had showed just a few years after 2013 and a few years before we published our work in 2016 that 146 and 124, they showed destabilized promoter G quadruplexes. They showed that in an in vitro study. They took out the gene, that region, and showed that in experiments that it actually destabilizes the GGG repeats in a test tube. So we started asking the question in a different sense. If these are G4s and if these are the G2A mutations, is TRF2 binding that we saw on the promoter of telomerase, does the binding require that there is a quadruplex present inside the cell? So how would you go about asking this question? So this became our question. First was to show that in vitro studies with recombinant purified TRF2 and a sequence that forms the quadruplex, and then you build those mutations in there, and then you show that the TRF2 likes to bind to the quadruplex forming sequence much more avidly, at least twofold higher, 2 to 3 folds higher than affinity than the mutant. So we asked, so it's still, could TRF2 mediated telomerase repression, could that be lost due to loss of TRF2 binding on the mutant promoter? So we needed to now go inside the cell and find out these issues where we have this mutant promoters. So there were one U87MG is a glioblastoma cell line which has that 1-124 G2A mutation. So we had that, and we saw that in this 124 G2A mutation, you will notice that the TRF2 occupancy is depleted. We don't see the TRF2 chip did not work in this cell line. TRF2 chip was negative. There was no TRF2 occupancy in the promoter of telomerase, unlike the normal case and unlike the cases where we did not have the G2A mutation. Similarly, LN229 is another glioblastoma line with a G2A mutation. There also we found the same thing. The occupancy of TRF2 is low. Now what we did is we had this cells from Vinay's lab, Vinay Tergaonkar's lab, who had recently, just a few years back, published a paper and where he had talked about this mutations in a very different context. But what was interesting is he had these pairs of cell lines where with or without the mutation. So HCT116 with the mutation, where he had created without the mutation, where he had created the parallel mutation 146G2A, BLM with the mutation where he had corrected it. So now we had pairs of cell lines with or without the mutation, with where otherwise the background was isogenic. So this he was very kind to share with us, and we were able to clearly see that as soon as you have the mutation, TRF2 occupancy fall on in the promoter falls down drastically, as if one single mutation and the whole promoter has lost TRF2 occupancy across the promoter. And that is not only true for this, any cell line, all the three cell lines that we got from him, pairs of cell lines, showed the same. TRF2 occupancy falls when one has this G2A mutation. And that would explain why telomerase goes up, because TRF2 is a repressor. So in the case of mutations, the occupancy is lost and therefore one can obviously, So we also noted that 27Me3 goes away and therefore the activation happens. Now, the next part was something very keen that we very anxiously wanted to study, and we went to we collaborated with Deoprakash Pandey from University of Oslo. The reason was they had patient cells from patients who had donated their glioblastoma patients who had donated their brains, and they had primary cells where these mutations were present in glioblastoma cells. So we had either a patient with a wild type promoter that is labeled here as G144. G7 was a patient who had the 120 minus 124 G2A mutation. G166 was a patient who had minus 124 G2A mutation, and G14 was someone who had minus 146 G2A mutations. And we immediately noticed that, as expected, telomerase activity was high in the case of the mutations. But for us, the most important experiments were the following. We were expecting that with the mutations, there would be loss of TRF2 occupancy from the promoter. That was the molecular aspect that we were most interested in. And it turned out to be so. As soon as these mutations were built, we took these primary cells, and once we started doing the TRF2 chip, we could see that with this point mutations, one single mutation, and as we noted with the previous cell lines, in primary patient-derived cells also, the same thing was very obvious and predominant. TRF2 occupancy falls drastically. Not only does it fall, the TRF2 occupancy for brevity of time, I'm not showing most of the data. We also saw EZH2 occupancy, 27Me3 occupancy, all the dependencies that we had discovered using the other cell lines were true for the patient cell lines also. TRF, in the case of mutations, when TRF2 doesn't come, it doesn't bring RE1 silencing factor rest, it doesn't bring the PRC2 complex, the 27Me3 mark is also lost. This was where I think we had a lot of debate that in case in case TRF2 is binding to G quadruplexes, what we were proposing, can small molecules which bind to TRF2, which bind to the quadruplexes specifically, and we had a lot of different kinds of quadruplex binding molecules sitting in the lab made by people across the world who have kindly shared this with us for the work that we have done for different aspects of the work. So we had this molecule sitting with us, and we started asking, how about stabilizing the quadruplex? One G has fallen off, the bookshelf is wobbly, the bookshelf is falling off, and once the bookshelf is falling off, TRF2 is not occupying. Is there a chance that you put in a molecule there which sort of props up the bookshelf once more, stabilizes the quadruplex, and the effect of a stabilized quadruplex would be return of TRF2 and therefore return of the PRC2 complex and 27Me3 and reclosing of the telomerase promoter. So this was a big question. This was a big question, and we hoped, anxiously waited for these experiments. And what we noted was this: SMH1 4.6 or JD83 are small molecules, which are published small molecules, which bind to quadruplex with 100 to 150 fold higher affinity than any duplex. And what we noted immediately was, in the presence of these molecules, this aggressive cell lines stopped their telomerase activity, which was otherwise very high, decreased significantly. Not only that, we also noted that gene expression, not the telomerase activity, the TERT gene expression, that is the activity of TERT, and this is gene expression of TERT, also significantly decreased. And the next part was, we had already known that, in this cell lines, the TRF2 occupancy had fallen, right? The TRF2 occupancy was lost because of the mutation. So in the presence of the molecule, can one see increase of TRF2 as if the quadruplex has been restabilized, and therefore TRF2 is invited back to bind to its to the DNA structure that it likes to bind to? That happened to be the case, and this was surprising. Though we wanted to do this and we're very anxious about the experiments, but it was very interesting to see that in the presence of these molecules, TRF2 occupancy is regained, not only in this and even 87 mg, but also 27me3 comes back, because as soon as TRF2 comes back, the rest of the steps sort of start happening. This is only to show that 27me3 and the telomerase expression therefore goes down in the presence of quadruplex stabilizing ligands. Mind you, I'm talking about 2 ligands. We tried about 15 ligands. 3 or 4 of them worked. 1 we are patenting, therefore it's not here. 2 molecules we have shown and published in a recent paper. But it works for about, out of 15, this worked for about 5 molecules, not all of them. So here is the scenario. This is telomerase promoter as it should be, and the reason I'm showing a dimer, because TRF2 works as a dimer, and therefore this is probably the scene that the quadruplex is this blue and red. Red are the quartets, and then this blue things are the bookshelf structures that I was talking about. TRF2 binds to them, rest comes on, and then the PRC2 comes. This is how it should be, because this is how you keep the telomerase promoter really tightly closed, and therefore not let the normal cell present a tumorigenic scenario. However, this is how telomerase is kept down, because 27me3 is there to close the in a pathogenic scenario, in a cancer cell, what we note is this: because of these 2 mutations which disrupt the quadruplex, right? This is my way of showing a disrupted quadruplex, and so you do not see TRF2 binding, and therefore telomerase is up, because if TRF2 doesn't bind, the rest of the thing doesn't happen. And what happens in the presence of small molecules, which are G quadruplex stabilizing ligands? What you see is this goes there, it stabilizes the ligand. This is the model that we like to believe. It brings back TRF2. Once TRF2 is back, we have RE1 coming back, and we have the PRC2 complex coming back, 27me3 coming back, and telomerase expression is now, again reined in, or closed because of the closed promoter. So this was mechanistically what we wanted to propose and show, and also bring to fore all the aspects of a weakness in a system, in a very robust system, a weakness, targeting that weakness using a small molecule, and at the same time saying that small molecules are generally talked about. Small molecules bind to proteins, and these are drugs, right? Here we are talking about small molecules which bind to a completely different type of target. They are binding to DNA structure. So it brings to fore a very different kind of molecular biology, very different kind of targeting, which is not protein and small molecules. This is DNA and small molecule targeting. And I will stop there. I've talked about most of the work of Anand and Shalu, and the rest of the few people are here. I'll have a little bigger than that now, but most of the others who contributed, I think, are here. Antara, Sulochana, Ankita, Saujanya, and Shubro. And of course, Deo from Oslo, and Geoffroy Roux from Paris, and Jerry Shea has helped a lot in terms of, Jerry Shea is a big name in the field of telomeres, so he was very kind with his comments, with his inputs, and it was a real big help when we approached him with our findings, especially on the telomeric findings that telomeres can, have gene expression across the genome. So he was very kind in looking at the papers and giving his inputs. Dave is a friend and a collaborator from Mayo Clinic who has been with us for many years now. Most of the funding was from India Alliance, DBT Welcome, and of course Council of Scientific and Industrial Research, where I work. And thank you very much. I'll be happy to, if there are questions, I'll be happy to talk more. Thank you.
Thank you so much, Professor Chaudhary. It was really the enlightening talk. Actually, the example which you have put, how to actually be a good teacher, means you elaborated this whole complex thing in such a simple words that it is really inspiring for me, for all the audience who might have heard you, and all the students who will learn from your talk and will be motivated to do similar kind of very hardcore epigenetic work which you have just elaborated. And at the end, you basically mentioned about the small molecule, which is not only, which is mostly people think that it is targeting protein, but you have shown that your molecules are targeting the DNA structure, which is also fascinating. So I think Alpana can help me in getting the question which must be coming on YouTube. Just to start with, I have some naive doubts which you which you may find very naive, and you please help me understanding. So basically, you have elaborated the mechanism of a tumor suppressor kind of situation, right? There is a structure change in the DNA which is not allowing this TRF2 to bind. So basically, the TRF2 function is lost, and then you are basically repairing the tumor suppressor kind of function. So which is very hard actually in the cancer biology. Regaining the mutation, which is basically healing those mutations, which is of tumor suppressor kind, is very hard in my belief. So please elaborate your success story on that, if possible.
In some sense, yes, it's right. It is in a larger sense, it is a tumor suppressor situation because TRF2 keeps telomerase closed, telomerase expression repressed, and keeping telomerase repressed is definitely a tumor suppressor function because high telomerase almost always, almost 100% of cases, telomerase reactivated telomerase leads to cancer. In some cases, it is telomerase independent, which is 95% of cancers are because of have telomerase reactivation. In 5% cases, there are cancers which do not have telomerase reactivation. That is also known. So telomerase reactivation being a cancer promoting scenario, any situation which keeps it repressed is a tumor suppressor scenario. So in that context, I think TRF2 binding to the telomerase promoter and keeping telomerase shut is a telomerase repressor, is a tumor suppressor situation, akin to tumor suppressor situation, because the textbook definition of tumor suppressor is a very different. In a larger sense, this is tumor suppressing event.
Yeah, not in the meaning of the textbook kind of definition, but yes, the situation is like that.
It's not a canonical canonical tumor suppressor situation. Let me put it that way. And so these mutations actually are creating havoc because it's very interesting. What I did not get into is telomerase promoter is the only promoter which has so many guanines in such a small sequence. So it has probably 15 different quadruplex forming potentials, different types of quadruplexes that it can form. And it's a very interesting question. Why would a promoter have so much quadruplexes, and this gene actually makes telomeres, which are again quadruplexes? So there's something very interesting there, and very perplexing also. So, and a couple of mutations which disrupt these quadruplexes create havoc. In glioblastoma, uh, it is, it is striking to see that one or two such mutations and what devastation it can cause, right? One single mutation, 1 minus 124 mutation, is
Right.
found in almost 35% of glioblastomas. And everybody knows what is the devastation how devastating that is. Yeah.
Thank you. So, are you elaborating this to other promoters also? Like you said that the G quadruplexes are there across the transcription start
Yeah.
site or near the promoter.
So yeah, so the first one that we showed as the first transcription factor that we showed was the MYC promoter, CMYC, which is another oncogene. There we were able to show that the transcription factor was important for, that's a very different transcription factor, not TRF2. It was a factor called NME2. NME2 is the first discovered quadruplex binding transcription factor, and we showed that it is important for activation of MYC, CMYC, and these mutations cause any mutation, not mutation. You can make a mutation in the CMYC promoter, and therefore see that NME2 doesn't bind. So that was the first example that we showed. After that, we showed the same event happens in the P21 promoter. P21 is a very important cyclin dependent kinase, which is activated following DNA damage response. There also we showed that the quadruplex is important, and across the community, I think now many more promoters have been shown which depend on quadruplex and cognate transcription factors for transcriptional activity. In a sense, what it is promoting is beyond the textbook understanding of duplex DNA. It is showing that DNA structure or non-duplex DNA also elicits function. It has function whereby, so DNA in some sense is beyond storage of information. It is also going into the realm of function. Specifically, what is important and interesting to us is the role that we see in epigenetic functions of this structure.
Sure, sure. Professor Arindam Mitra would like to ask you something, or he has some comments.
Sure.
Shantanu, first of all, many thanks. I'm sure Sandeep has a formal vote of thanks, but let me thank you for quickly accepting our request and delivering this wonderful talk. I mean, I was mesmerized. I also recollected some of the earlier days in ITIB when you started this G quadruplex work and we had some chat. I have one question though, question or a comment. So we are also interested in telomere hemostasis in some other aspects of human health. And we know that this telomere hemostasis is like a dynamic equilibrium. It gets depleted, the length gets depleted as more and more cell divisions happen and gets repleted by the telomerase, and particularly what you showed here. Now, did you by any chance see whether after adding these inhibitors, small molecule inhibitors, ideally you are actually disturbing that equilibrium in the sense that now the cells are going on for multiple divisions and there should be a reduction in telomere length, which is these days we can assay by genomic assays are there for that, which is like real-time PCR. So were you able to see that? I mean, I was just curious.
Yeah. That happened to be a very important question in the lab because we follow telomere length in different ways. We look at telomere length heterogeneity in different, So yes, as soon as telomerase is repressed, within a few cycles, you see telomere length going down. And that is how the cells which had lost their molecular clock Now, once telomerase is repressed, the cells become trigger the apoptosis, and this very aggressively growing glioblastoma cells, in the presence of these small molecules, within 4 to 5 divisions, started becoming cytotoxic and started dying.
Oh, that quick.
Yeah, so it just takes about, because this, what happens in these cancer cells are telomeres decrease to a certain length and then are maintained. So as soon as you decrease telomerase, it very soon goes beyond the critical length. And as soon as the critical Hayflick's limit is crossed, the apoptosis DNA damage response is triggered and apoptosis takes over. So yeah, to begin with, therefore, when we use these molecules to begin with, we could see telomerase going down, but the cells remained happy. So those are some anxious time for both Shalu and Anand. Nothing is happening to the cells, but it took about 4 or 5 divisions before we could see the cytotoxicity kicking in. That is exactly because of this reason, because telomeres start decreasing. It takes a few cycles for the telomeres to decrease to the critical limit, below the critical limit.
Thank you. Thank you so much.
So you need to keep your molecule on the cells, or it is like a just one-time treatment and then 4 or 5 cycles can be taken care?
I think we had to give two times, and we could see that after 3 days we had to give it again. Yes.
So means the new DNA needs it again to be blocked.
Yeah, to be blocked. Yes.
Okay. So if there is no more question, Alpana, do you have any question on the YouTube? Okay, good. So maybe people will watch the video again and again and then put a lot of comments. So thank you, Professor Chaudhary, for giving us giving us time for your distinguished lecture today, and we will love to have you here in person, and we'll find the opportunity to invite you again in person in the campus as for your input. And yeah, so this is unfortunately that we are doing it online, but we would have loved to have you here in the campus. But thank you so much for giving us your time and putting this talk in front. And I will also thank our NIBMG faculty members, and especially the IT section, for keeping this infrastructure so that we could actually deliberate your talk, or you could deliberate your talk on online platforms. So thank you, Bikram and his team, for his effort to make this event successful. So thank you, Professor Chaudhary, and thank you, Professor Mitra, for joining us. Thank you very much.
Okay. Bye-bye.
Bye-bye.